Welcome to this special episode of the Powerhouse Podcast. My name is Dr. Keri-Lyn Kozul, and I am honored to be on this journey with my co-hostesses, Hannah Pletcher, a graduate student and science communicator, and Geoghegan, a Mito mom and advocate for families in the Mito community. Today, we're taking you through the story of FBXL4-Associated Mitochondrial Disease, a rare mitochondrial disease that affects children and families across the globe.
There are a lot of terms you'll hear throughout this episode. When we say FBXL4-Associated Mito, we're talking about the disease. And when we say FBXL4 protein or FBXL4 gene, we're talking about the protein or the gene, so more on the science side. This episode is part history, part science, and part family stories. You'll hear from the researchers who made key discoveries, and you'll hear from patients who live with this disease every day.
And I'm here representing the families. Both of my children, Lorelei and Benji, were born with FBXL4-Associated Mitochondrial Disease. Lorelei is no longer with us, but her life was full of light and joy and love despite her disease. And Benji, my little bear cub, he is still here, still fighting and reminding me every day that these kids are incredible and children first and patients second.
So let's dive in. Mitochondria, the powerhouses of the cell, are the batteries of our cells that make energy so our cells can function. In mitochondrial disease, or mito as we call it, these batteries don't work properly, so the body doesn't have enough energy.
FBXL4-associated mito is incredibly rare because it is a uniquely different type of mitochondrial disease. Basically, our cells have a way of knowing where their mitochondrion battery is not working. And so they have these ways to replace and remove them if they're not working. For the science people out there, this process is called mitophagy or mitophagy. In the case of FBXL4-associated mito, the disease mechanism lies in this removal process. So our cells know to only remove damaged mitochondria because we have this protein called FBXL4 that regulates the removal process. FBXL4 protein helps keep mitochondrial removal levels low. But when FBXL4 is mutated, like in FBXL4-associated mitochondrial disease, for example, FBXL4 protein cannot keep mitochondrial levels low, and so too many mitochondria are removed. This leads to patients not having enough mitochondria to function, and you can only imagine how devastating that manifests.
FBXL4-associated mito is unique. While most mito is caused by dysfunctional mitochondria themselves, FBXL4-associated mito is caused by dysfunctional mitochondrial removal.
Exactly. You may have heard this type of mito called mitochondrial DNA depletion syndrome. With FBXL4-associated mito, too many mitochondria are removed, and this is what depletes mitochondrial DNA.
So cells without FBXL4 protein are removing mitochondria when they shouldn't be, because FBXL protein is what keeps our mitochondria around. What causes cells to not have the functional FBXL4 protein?
Dysfunctional FBXL4 protein is caused by a super rare mutation that both parents have to be carrying in order for a kid to inherit.
And the result, most patients present with incredibly severe symptoms that range from brain developmental defects to metabolic defects and everything in between. This is a multi-system disorder, and there's a wide range of potential symptoms.
Our goal today is to bridge the gap, to connect the work happening in the labs with the reality of families living with FBXL4-associated mito.
Let's start at the very beginning.
The story of FBXL4 as a diagnosis begins in 2013. Since we're recording this in the fall of 2025, this is only 12 years ago. That's when the very first case was identified, Juliet, by Dr. Falk's team at the Children's Hospital of Philadelphia. We were fortunate enough to chat to Juliet's mom, Janice, about her journey with FBXL4-associated mito.
So I am mother to Juliet. Juliet was diagnosed with presumed mitochondrial disease at a very young age. She was pretty much sick since birth, but she had a muscle biopsy at the age of 1. It did not give us a lot of information other than her cells, the energy wasn't being produced correctly. My husband and I, we decided that we were going to give her the best life possible. At the time, we were told she probably wouldn't live to be 5, which was pretty standard. And so we just made sure that we wanted to give her as many experiences as possible. Juliet was just sweeter than sugar. She was full of joy, even though her life was very challenging. She never walked. She was in a wheelchair. She never talked. She made some noises and a lot of laughter. But she never talked. She never ate by mouth. She was on the more severe end of mitochondrial disease. And so even though she had such a difficult life, she taught me so much about finding joy in simple things. So Juliet was the first child in the world to be diagnosed with FBXL4 mitochondrial disease after an 8-year journey to find that diagnosis. Juliet progressed in her illness, and she tragically passed away when she turned 16. It was 5 days after her 16th birthday.
Back-to-back publications led by scientists in the UK and the USA were published in 2013 about the severe symptoms that kids like Juliet had to face. The main symptoms being a loss of mitochondrial DNA and mitochondrial encephalopathy, which is a brain condition due to abnormal mitochondrial function.
Juliet's family spent 8 long years searching for answers. Finally, they were told that their daughter was the first known case of a newly recognized mitochondrial disease. Imagine being told that your child's condition is so rare it's literally never been named before. We asked Janice what that felt like.
I think the word that would describe it best is frustrating. It was frustrating because we knew so little about what was happening, even though she had had so much testing done, including ultimately her whole exome sequenced. And so every time Dr. Falk at Children's Hospital of Philadelphia would come close to what she thought was a diagnosis, it would be a red herring. So it was frustrating. The process was frustrating. But I knew, because we work so closely with all of the scientists at the Children's Hospital of Philadelphia, that there were so many brilliant minds working on trying to give us an answer for our child. And hopefully that answer would lead towards better, more customized treatments and potentially a cure. So it was frustrating in the process. But after we finally determined it was FBXL4 mitochondrial disease, we had a name for this thing, and being able to identify what the whole process is, and believe me, that's what the scientists are there for, gave us hope that we would be able to serve her better on the day-to-day, possibly slow the progression of the disease. In hindsight, we would love to have been able to do that, and it did not happen for Juliet. But knowing now that, a child who presents with certain symptoms, even a few days old, will be able to be identified so early and therefore receive hopefully more individualized treatments, makes me really proud of Juliet. And we're just so grateful for all of the doctors and scientists who have worked to help parents with that process. No, we knew that she was the first in the world. I mean, it was a pretty big endeavor that Dr. Falk had initiated and worked with other scientists around the world. So we were a part of the inner workings of it. So we knew it was something very, very unique. But what comes with being the first in the world is that we don't know much. So it was kind of like winning a prize that you really don't want. I mean, now the thought that so many children can be diagnosed at such a young age, it's rewarding, but at the time it was a little scary that she was the first. Like, what do we do with this information? And even now, we know a lot more about FBXL4 mutations and how it works, but there's still so much work to be done.
Hearing Janice, I can feel the mix of relief and heartbreak. As a parent, I can only imagine what it felt like. Relief to finally have that answer, but also the weight of knowing that there's no roadmap, no community, no treatment. But there was Juliet, a beautiful, sweet girl who was touching the hearts of every person she met. Janice shared her favorite memories with Jules with us.
I thought about this a lot. I talked about this with my husband, and we had kind of a unique situation at the end of Juliet's life because of COVID. And it sounds a little strange, but we had Juliet on hospice for about 10 months before she passed. And she was on hospice for about 6 months during COVID, which provided us with a really unique experience. As a medically fragile family, by the time Juliet was 15, we had 24-hour nursing care because her health was dependent on all of that. She had TPN, and it was challenging. And so COVID comes along, and now we don't want any medical professionals in our home because we're afraid they're going to bring in COVID. And we just fought for 15 years to try and keep this child alive. We did not want her to pass from COVID. So and I don't think the nurses really wanted to come in. Like, everything had scaled back, right? So the last 6 months of Juliet's life were just Juliet and me and my husband, and no nurses in the house, no visitors either. And because I win really strange prizes, a tree came through my house 3 months before Juliet passed, and it forced us to go to a house that we had in the Pocono Mountains in Pennsylvania, which we called Juliet's happy place. And so the last 3 months of Juliet's life were spent with just mommy, daddy, and Jules. And we sat on the water every day, and we took her out on boat rides. And I am so grateful for that time with her. So I don't know that there's one memory other than her being so joyful, but I feel very blessed that we had that.
Juliet's story also highlights something important. Even today, many mitochondrial disease families still don't have a genetic diagnosis. For some, that uncertainty stretches across years, sometimes even decades.
FBXL4 is just one piece of a much bigger puzzle. Every time we identify a gene, we open doors for research, treatment, and support. But there are still countless families searching for answers. Janice had a message to share with families awaiting a diagnosis.
The one message that I do think is important to note for any family that may be listening to this that doesn't have a diagnosis yet is that your day-to-day doesn't really change. You need to continue to advocate for your child medically and stay on top of all of the interventions that will help your child to, live their life to the fullest day to day. That isn't necessarily going to change. What changes is the hope that you feel knowing that so many brilliant people are working to help your child. So that really is the change, was the change.
In 2019, things took a turn for the better, and science began to uncover some of those answers. Researchers developed a mouse model for the FBXL4-Associated Mito, where the protein is knocked out and isn't functioning as it's supposed to. Researchers do this to figure out what a protein is important for and what goes wrong if the protein isn't there, like what happens in this disease. This is the first ever animal model of FBXL4-Associated Mito, which opened up a plethora of information for us.
This work was co-authored by Dr. David Alsina in Dr. Nils Larsson's lab at the Karolinska Institute in Stockholm, Sweden. We got to ask Dr. Alsina a bit about his work.
There was few research done on this protein. It was not known what the function was in the years before we published, since let's say between 2013 and 2018, there were more and more reports of patients coming up. So we thought it was a good candidate to generate mouse models and to explore what this protein was doing.
David and his team's work in generating FBXL4 mites uncovered some key defining features of FBXL4-associated Mito. Let's hear from Dr. Alsina more about his findings.
So the key findings of our manuscript is that we basically found what is wrong with these patients' cells and why they become sick. And what we found is that in the absence of FBXL4, or when this protein is mutated, mitochondria are degraded in a very fast way, in an uncontrolled way, we could say. And so there is very few mitochondria left in the cells of these patients, and they cannot produce enough energy so that these cells can work. So in a way, the mitochondria don't have a problem in producing energy, but there is too few mitochondria to sustain energy production in these patient cells. And I think that's the highlight of our manuscript.
So initially, we knew that FBXL4-associated Mito patients had less mitochondrial DNA, but David and his team found out that there were less mitochondria.
Yes. So for our non-sciencey people, you're probably wondering why this is a big deal. And I can break that down for you. Mitophagy is the scientific term for the way our cells get rid of mitochondria they don't need. Think of it like this. Mitochondria are the batteries of our cells, and they make energy when we need to function. But when batteries stop working, we remove them and replace them with new ones, right? Turns out our cells do this too. And different things trigger why we remove mitochondria. Sometimes it's because the mitochondria is broken. Sometimes it is in response to different signals like less oxygen or iron. And sometimes cells get rid of mitochondria as they go through different developmental stages. David and his team uncovered that the FBXL4 mouse has elevated levels of removing mitochondria.
So basically, since that publication in 2013, which included Juliet, we knew that these kids have less mitochondrial DNA, or mtDNA. But David's publication was the turning point that helped us identify that there are actually less mitochondria overall. So this is why dysfunctional FBXL4 proteins leaves kids without the energy that they need.
As a mom, it's powerful to hear that scientists talk about all of this happening at the cell level, because it connects back to what we see every day in our kids' lives.
By 2021, FBXL4 was no longer just a mutated gene on paper. Families all around the world were navigating life with this diagnosis and loss.
2021, that was a big year for my family. That year marked both devastation, loss, and ongoing hope.
Yeah, we sat down with Susan and her husband Michael to learn more about their journey with FBXL4-associated Mito.
Do you want me to start?
I guess I can start.
Okay.
So our journey, I guess, starts in 2016 with FBXL4. Initially, when Susan was pregnant, we had no idea anything was wrong until later on in the pregnancy. I think it was like Super Bowl weekend. We were at a friend's house and her ankles were swelling up, and then I think it was that week she went into the doctor's office, and then she called me crying that they were admitting her to the hospital for preeclampsia. So that's kind of where it started, and then
It snowballed.
Yeah,
very quickly
through ultrasounds. They're like, okay, something else is wrong, and we were transferred from Riverside to Norfolk General, which has the
It's next door to the Children's
Yeah.
Hospital of King's Daughters.
Yeah.
They wanted us close to a
the NICU. That's what
level 4 trauma NICU so that they could, in case something was wrong. Go ahead. Sorry.
Yeah. So then she gets there, and then we find all of a sudden they need to do an urgent C-section. So they do the C-section and take Lorelei away to the NICU to work on her. So didn't even see her really initially.
And I know that's different from a lot of the other FBXL4 families that we've met. A lot of them had their babies and they didn't know something was necessarily wrong, but they had time with their children. And Lorelei came in.
And she was premature. This was 32 weeks, some odd days, and then very small for 32 weeks. She was 1 pound?
She was
10,
2 pounds, 13 ounces.
Okay. Yeah.
So halfway through our NICU stay, actually, the really cool thing for Lorelei doing holidays the way she did, we got her diagnosis on Rare Disease Day. So, little nod to Lorelei, but sitting in a conference room, that was the first time we heard mitochondrial disease. And they didn't have much information at the time. There were only 2 or 3 case studies published total. And so they believed that Lorelei was the 31st documented case. I thought they meant at the hospital, but they meant like total, based off of how few case studies there were. So we did a NICU stay and left the NICU after 77 days. We had to learn a lot. Like, neither of us have medical experience. Neither of us have a science background. So it was a lot of learning. I always joke that like what I knew about medicine was whatever I picked up watching Grey's Anatomy. Like, that was the extent of my medical knowledge. So that she was born in 2016. And then in 2019, Benji joined our team. And Benji also has FBXL4 mitochondrial disease. It was a transition having both of them.
Yeah. When Lorelei was born, they had only discovered the role of FBXL4 in this mitochondrial disease in 2013, I think. So it was just 3 years prior. So I know there's a lot of families who go for years trying to find a diagnosis. We were lucky that some families before us had gone through and the researchers working on it and were able to identify that gene, because we got a diagnosis very, very quick there in the NICU. It didn't take long. After ruling out some other things, they did the whole exome sequencing, and it wasn't too long after that we got
13
the results back.
She was 13 days old when we got the diagnosis. And then with Benji, we found out through an amniocentesis, only because they knew what to look for because of Lorelei. So it wasn't something that would have ever have shown up if Lorelei hadn't come first. And when he was born, he presented very differently than her. We made it to term with him. And I always kind of joke that between the 2 kids, we'd check off all the clinics and departments in the hospital. Like, she had more neurological complications. He had more cardiac complications. Her eyes, his ears, like, it just kind of seemed to work that way. So we had some scares in the beginning with him and his heart, but our team was on top of it. And now his heart is very stable, knock on wood. And yeah, it was a lot of learning and a lot of juggling. In 2020, we all hunkered down like the rest of the world. And it was probably the best year for us because we all got to be home. And it was that like perfect moment. I think we talked to Janice and she said 2020 gave her so many blessings, and it did for us too, because we were all home and like everybody was stable. It was the only time with both of my kids here that everybody was good. Lorelei had some shunt complications at the end of 2020, and that kind of began her decline. She passed in July of 21. She was 5.5 years old. And yeah, it was really, really hard, but also just kind of, I mean, we're 4-ish years removed now, and it's kind of part of the journey. And I often wonder like how I would be doing this if both of them were here, because our children have such significant needs. I know FBXL4 is a spectrum, and our kids are on the more severe side of the spectrum, needing, they have feeding tubes, they can't walk or talk. Benji is using an eye gaze device so he can communicate a little bit with a Tobii Dynabox talker, which is really, really cool. And so he can tell us a little bit like, I want to go play baseball and things like that. But it's been a very unique journey. And yeah, now he just turned 6, so he outlived her, which is incredibly bittersweet. But he's writing his own story.
I think it's important to highlight in the Geoghegan story how powerful it is that kids like Juliet paved the way for quicker diagnosis for kids like Lorelei and Benji. Juliet's family, the Belchers, waited years for diagnosis. The Geoghegans waited days for Lorelei's diagnosis, and then Benji was diagnosed before birth.
That's really true. Sometimes it can feel disheartening that we don't have cures despite identifying these types of rare mitochondrial diseases and doing research on them, but identifying these rare mutations are incredibly beneficial in making quicker diagnoses and working towards treatment.
Yes, and amidst the journey, the storms that life throws, warmth comes through these kids who remain so special and important to their families. Suze and Mike shared their favorite memories with their kids with us, and it was really special to hear.
Favorite memory is just how happy she would be, even under difficult circumstances. I like distinctively remember her in the hospital in the PICU when she was having a shunt revision done, and she had the external. I don't remember what they call it.
Brain drain.
Yeah. The external shunt, because hers was infected. And even with that in the hospital, just her laughing and smiling through it all.
And when I was preparing for this, trying to think about a favorite Lorelei memory, immediately I go to like Disney and like her surprising me and like weight bearing, standing with the princesses. But then I was like, no, like that's not my favorite. My favorite memory is like the little moments, like her just laughing hysterically at home. A few days before she passed, I have a video of her laughing at Alexa playing fart noises, and she is just hysterical. And just, she was such a good cuddler. So like, it's the small things that are actually the big memories.
And then I'll go.
Benji and Lorelei are very different. I actually kind of chuckled when I saw the question, because Lorelei was a sassy, bubbly, funny thing, and Benji is so just sweet and calm and so serious, and he loves so hard. Benji loves his mama and his dada and his blankie, and the things he loves hard, where Lorelei loved everything. But Benji also, he plays baseball in his wheelchair, and he loves his movies, and he's a fun, quirky little thing. What were you gonna say?
Benji is very different than Lorelei, and I think a lot of people feel sorry for the situation that you're in, but it's the small things like you said before, and even just watching him lie there sleeping at night just makes you feel lucky and blessed to have him.
Hearing Suze and Mike's journey shows us the full weight of what FBXL4-Associated Mito means, the grief of losing Lorelei, and the hope and resilience that we see in Benji.
And families like theirs are why the research matters so much. It's not just data, it's children, laughter, love, and memories that deserve to last lifetimes.
The FBXL4 parent community has grown into a vital network of support, and organizations like the Apricity Hope Project and the When Autumn Comes podcast, both founded by Suze, are connecting caregivers and giving families a voice. Suze told us more about her incredible work supporting caregivers.
I started the Apricity Hope Project, and one of our programs is the When Autumn Comes podcast. And a lot of people think that I started it because of my kids, and really I didn't. I started it because of the version of me that sat in the NICU and got the really bad news and was terrified and had support, but not the support I needed. We are very lucky because our friends and family are very like, what do you need? We've got you kind of people. But early in the journey, especially watching all of my friends and their kids do what typical families do, and watching all of them like just do the things that we realized we were never going to do with our kids. It is a very isolating journey. And then add the fact that both kids are immunocompromised. We can't really go and be around a lot of people. So I started the Apricity Hope Project. Apricity means the warmth of the sun in the winter, because I know that this is a very cold, dark, isolating season. And AHP wants to be the light and the warmth for caregivers. So we do retreats and meetups. We have moms come in from all over the country to our Hot Pink headquarters here in Norfolk, Virginia. And we do 4-day retreats. We do hospital go bags that are for parents. They are not, there's nothing in these bags for children. It's all support items for parents who are in the PICU. We do NICU graduation bags to kind of capture that version of me as she was getting ready to leave the NICU with a brand new Lorelei and a lot of questions. We have support in there for them so that they can find community immediately. And then we also have a branch that helps. We're growing it right now, but we've always given money to mitochondrial disease research. And now we are focusing it a little bit more and kind of working on the community of FBXL4 families and creating like welcome to FBXL4 packages that kind of have info kits for them. And we have stuffed bears that Build-A-Bear donated to us that we call Benji bears, and they can go to the kids, and just trying to be the face of this disease for families, because when you Google it, it's terrifying. There's no good news out there. And I feel like a lot of what gets lost, not just with mitochondrial disease, but with all rare diseases, is that our kids become, the disease. And when you Google it, you never see happy children. You always see kids in really tough times. And I guess I really wanted families to feel like just because you get a diagnosis doesn't mean the story is over. It just means the story is different. And I want to encourage families to know that, like, I always tell everybody, like, this is going to suck. It's going to be really, really hard, but you can do it. It's just going to look different. And yeah, so AHP is here to support families, primarily moms, but the dads are starting to trickle in, and we've had one dad's retreat, and we're going to do more. We also offered our first bereaved medical mom retreat because I feel like once a medical mom, always a medical mom. We carry lots of trauma and baggage with us, even after the fact. So having one kid on earth with me and one kid in heaven has given me a really unique perspective to be able to care for both sides of the community. So yeah, I love it. I started When Autumn Comes in 2020. It was just a way for me and a bunch of moms to, like, we were already talking in the middle of the pandemic, so why not record our conversations? And we started recording. It was me and a mito mom from Minnesota who I had never met before. And we started co-hosting, and We've now released over 120 episodes and 100 pieces of bonus content. And I now have a co-hostess, Katrina from Canada. Her daughter has a different form of mito. And it's a really cool way to connect people. And it's not just mito families, it's all families, because really we all feel very similar emotions, grief, trauma, heartache, but also the joy. When autumn comes, Diane was my co-hostess at the time. She's from Minnesota. So with her accent, when we took the word mito, M-I-T-O, and when you spell it backwards, O-T-I-M, it sounds like autumn if you're from Minnesota. So we played off of that. When autumn comes, when mito comes, it's kind of the roundingness of the darkness is coming. But it's also, there's golden leaves and beautiful sunsets and perfect mito temps for my kids. So there's joy in the midst of all of it.
Suze, thank you so much for doing this. I'm truly really inspired by your commitment to support others with your unique perspective and experience. And it's really making a difference in the lives of so many families.
Yeah, my hands were tied when it came to the science side of this disease, but I'm really good at connecting families and resources and helping them feel less alone in their world when it is inevitably going to flip upside down. In order to care for our children, we have to remember that we have to care for the parents too. And this is a team project. We are all doing this together.
Suze, you're also an excellent up-and-coming novelist, right?
Really?
I can't believe you guys put this in here.
Yes, Hannah, you've got to read Susan's book. She has been generous enough to share her story as a mom, caring for and raising sweet kids who have had to journey through mito. I was lucky enough to pre-read it, and trust me when I say it was one of the most beautiful manuscripts I have ever read. And this story will resonate with me for the rest of my life. Where can listeners access your book, Susan?
I can't believe you snuck this in here. My launch day for Live Like Lorelei is October 10th. This is what I get for not doing my homework. You can grab a copy on Amazon anywhere in the world. I think that every parent with a kid who has mito has their own unique journey, but sharing our stories helps make it all a little bit easier. You can feel less alone. It's less isolating when you can see yourself through someone else's story.
Well, Suze, you should take that as a very high compliment that Keri said that it's one of the best manuscripts she reads. She reads all of our manuscripts, so this is a big deal. And I'm really, really excited to read your book. And it's so kind of you to do so much for the community. This community is also a bridge. It connects families to the science that can feel so overwhelming.
Well, we will share details for the Apricity Hope Project, the When Autumn Comes podcast, and also the Love Like Lorelei book in our show notes, because I would highly recommend you check them all out. But we're going to take a quick break right now, and we'll be right back soon.
We hope you're enjoying the episode. The United Mitochondrial Disease Foundation's Mitochondrial Medicine Conference brings together researchers, clinicians, and patient families to work toward better outcomes for patients with mitochondrial diseases.
Save the date and make plans to join us in sunny Orlando, Florida for Mitochondrial Medicine 2026. Visit us online at umdfconference.org. Now let's get you back to the Powerhouse.
Welcome back, listeners. The UMDF MitoMed conference is genuinely my favorite conference of all times. And I always look forward to meeting new researchers and patients. And I have so many stories from my experiences there. So if you have the time to come out to Florida next year, we would love to meet you. And I think it would be a really good opportunity. I'm going to try my best to be there. We appreciate you joining in on our FBXL4 Powerhouse podcast. But now, back to business.
Fast forwarding 2 years in 2023, the tide shifted. A flurry of research papers were published, each adding another piece to the FBXL4 puzzle.
These studies revealed new insights into the molecular mechanisms of how the disease works, meaning what's actually going on inside of the cells of patients with this disease, and uncovered the first ever known role of FBXL4 as a protein.
And for families, these findings meant something very, very simple: progress, hope that science is catching up to our kids' stories.
This work was the foundation of my PhD, and one of the 2023 publications was my first ever publication. I did this work in Dr. Juliet Pagan's lab in Brisbane, Australia, if you can't tell from my funky accent. And we got to chat to Dr. Pagan about the exciting breakthroughs we made in 2023.
Okay, so the goal of our 2023 EMBO paper on FBXL4, we started by wanting to understand a process known as mitophagy, which is a cellular cleanup system where damaged or excessive mitochondria are removed from cells. So mitophagy has to be very tightly balanced in the cell. If it's deficient, then damaged mitochondria will build up. That means that the mitochondria will be leaking energy, harmful byproducts, and could contribute to disease. And on the other hand, if mitophagy is excessive or too much, then the cell will remove too many mitochondria, even ones that are healthy and useful, so that will leave the cell unable to function properly. So in our manuscript, we found that a protein called FBXL4 plays a key role in setting the balance of mitophagy. And it does this by controlling the levels of two of the mitophagy on switches called BNIP3 and NIX. They activate mitophagy. Without FBXL4, these switches will stay on, mitophagy will be excessive, and the cells will lose too many mitochondria. FBXL4 is mutated in mitochondrial DNA depletion syndrome. So these findings suggest the mechanism contributing to that disease. It had previously been identified as the causative gene by the groups of Penelope Bonin, Robert Taylor, and Massimo Zeviani. And more recently, Niels Larsson's group showed that FBXL4 deficient mice lose their mitochondria through autophagy. So our work, and together with the work of 3 other groups who published concurrently, outline the mechanism by which FBXL4 could be contributing to these diseases. So we found that the pathogenic variants in FBXL4 are no longer able to sufficiently suppress mitophagy. So that is why the cells lose too many mitochondria, draining the cell's energy supply.
So, Keri, your work with Dr. Pagan found the reason that the FBXL4 mouse model had high mitophagy. FBXL4 protein was regulating mitophagy to keep it low and save mitochondria. But when FBXL4 gene mutations were modeled, it could no longer keep the mitophagy low.
Yeah, so FBXL4 protein is kind of like a cell superhero. It protects mitochondria at all costs by removing the proteins that signal for mitochondria to be removed. The best part was that our group wasn't the only group to find this out. Dr. Pagan commented on the importance of collaborations in doing science.
An exciting part of our work in the last few years has been collaborations with groups from all over the world. So in Australia here, our groups worked with teams in Newcastle, UK, Liverpool, the USA, to figure out how all these mitophagy activators and mitophagy suppressors work together to suppress mitochondrial disease. For this, we have all engaged in communication that is open and reached out for advice and to share insights to make sure we don't duplicate progress and really streamline the research. It's this kind of collaborations that I think are really important for driving research forward for rare diseases. We've also realized that there's people all over the world working on research and coming up with the same results that we're having in Australia, in China, in the US, in England, and it's been a really fulfilling to realize that our results are robust and reproducible, and that other people have the same ideas as us and are finding the same results.
How cool is it that 3 labs literally on different sides of the world were able to confirm the same thing? Together, these discoveries gave us a clearer picture of what FBXL4 protein does. And now we have these clues to work with and a more targeted approach for how one might one day be treated.
And again, for families, it means hope, that science is moving, that our kids' stories aren't being forgotten.
So let's take a step back and summarize what all of this means. FBXL4-Associated Mitochondrial Disease presents with a range of severe symptoms. Canonically, it presents with a depletion of mitochondrial DNA. However, we more recently have found that whole mitochondria are lost. Basically, cells have evolved a way to target damaged mitochondria for removal. This process is known as mitophagy or mitophagy. Mitochondria have receptors, BNF3 and NIX, which signal for mitochondria to be removed. But to protect healthy mitochondria from removal, FBXL4 degrades BNF3 and NIX. This preserves the healthy mitochondria to produce the energy that we need to function. However, in disease, healthy mitochondria are removed, and this is because when FBXL4 is mutated, BNF3 and NIX accumulate and signal to remove healthy mitochondria, meaning that there are less mitochondria to produce energy, subsequently causing disease. Uncovering these mechanisms were a huge development for us. Before that, we didn't even know much about regulating BNF3 and NIX. During my PhD, I presented this at a mitochondrial disease conference, and Dr. David Pagliarini from WashU was in the audience and connected some of the dots. He knew Dr. Natalie Nemi, who was also working on a protein that potentially regulated BNF3 and NIX, except it wasn't FBXL4.
Dr. Nemi has been working on a protein called PPTC7, doing something in cells that was very similar to the FBXL4 protein. We chatted to Dr. Nemi about her work on PPTC7.
So our lab has long been interested in how mitochondria are regulated and how they adapt to different conditions and different physiological contexts. And we think that a process called phosphorylation might regulate these mitochondrial functions. So phosphorylation is a very fancy term that essentially means that there's a small tag that's added onto a molecule, like a protein or a metabolite, and the tag can change the function of the molecule, either the protein or the metabolite. So you can think of it as kind of an on-off switch. If something has a tag, a phosphorylation event on it, perhaps it turns the protein on. And the nice thing about this is the tag can be on for a short amount of time. So we can tag something to turn on a protein or a pathway, and then we can take the tag off and essentially shut that pathway down. So it's a very rapid and reversible way to kind of control and fine-tune mitochondrial functions. So one of these enzymes that kind of controls the tag, whether the tag comes on or specifically if the tag comes off, these enzymes are called phosphatases. And PPTC7 is a phosphatase. We know this because it looks like a phosphatase when it folds. So our initial goal was to try to figure out what PPTC7 was doing in terms of putting tags onto proteins and taking tags off of proteins. But we had this really unexpected surprise about this gene when we knocked it out in mice. So essentially we could use genetic tools to be able to precisely get rid of just PPTC7 expression and have everything else intact. And when we do that, the mice die within a day of being born. So they're all born, but this is a very severe phenotype, much more severe than we were anticipating. And what this tells us is that PPTC7 is important, if not essential, for development in mice. So we wanted to know why. And for many years, we assumed it was because PPTC7 was a phosphatase. It was pulling these phosphates off of certain proteins or molecules, and getting rid of that function is essentially what was causing this severe phenotype. But we and others really recently found that PPTC7 has a second function, and that is to negatively regulate this process called mitophagy. So mitophagy is essentially mitochondrial autophagy. It's the clearance of whole organelles, And it's a turnover mechanism, and it's thought to be able to quality control different mitochondrial populations to keep a healthy mitochondrial population. And so we and multiple other groups across the globe have actually found that PPTC7 is a negative regulator of mitophagy. So this is a really interesting enzyme in that it has at least two functions. It takes these tags off of proteins, which is what we were originally thinking we were going to study. But it's also a negative regulator of mitophagy, so it puts the brakes on this. So it turns out when we lose PPTC7, is what happens with these knockout mice, they can't stop the turnover of mitochondria. So the mitochondria turn over too rapidly. And if you have too rapid of turnover of mitochondria, you actually have a lower number of mitochondria in the cell. And we think this leads to an energetic crisis because there's fewer mitochondria to be able to make ATP or energy that the cells need to thrive. So we're still really interested in trying to understand this interplay between the phosphatase role of PPTC7 and the mitophagy regulation of PPTC7. We found it's very complicated, but that just means we have a lot more to learn.
Dr. Nimi was the first to study a mouse model of PPTC7. Removing PPTC7 protein from mice affects mitophagy, which sounds a lot like FBXL4 protein. We asked her whether she noticed similarities to David's FBXL4 mouse model.
They're fairly similar, FBXL4 knockout mice and PPTC7 knockout mice. So FBXL4 knockout mice, when you knock this out globally, meaning in all different tissues at the same time, most of the animals will die, but some of them will survive in the FBXL4 knockout animals. I think it's a pretty low percentage of the ones that survive, maybe fewer than 10% of them. But some of them do survive. And actually, once they make it past that first day after birth, they grow up to be reasonably normal in terms of when they're not stressed, they're not sick, all of these different things. In PPTC7 knockout mice, we've actually never had a knockout that has survived. So we've been breeding these animals for about 10 years. And we've never had an animal make it to weaning, meaning 3 weeks old. So it seems like PPTC7 has a slightly more severe phenotype, although the severity is fairly similar and the presentation is very similar. I think there's metabolic defects and mitochondrial DNA depletion because there's fewer mitochondria, so there's fewer mtDNA molecules around. So the mice are very sick. And I'd say there's a lot of parallels between the two, which emphasizes the fact that these two proteins probably work in the same pathway.
It's so fascinating to me that there are several families that I personally know of that are affected by FBXL4 mitochondrial disease, but I haven't heard of any that are affected by the PPTC7-associated mito. Natalie shares with us why she thinks that FBXL4 patients are more common than the PPTC7 patients.
It's hard to say because it's hard to comment on things that don't exist, right? It's kind of this bias of ascertainment. We don't really know. But the severity of the phenotype in the PPTC7 knockout mice would suggest that humans that have similar full loss of function PPTC7 mutants would be very, very sick. So it might be that that's why we don't frequently see them in terms of mitochondrial disease patients.
Dr. Nimi's work on PPTC7 led her to FBXL4 because in 2024, we discovered that FBXL4 protein may need PPTC7 protein as a helper to function in preserving mitochondria. She explained to us how both proteins are connected.
So we've known that BNIP3 and NIX, which are these mitophagy receptors, they are the things that actually recruit the machinery that's needed to turn over mitochondria. And it's been known, I'd say, for quite a few years, actually, that BNIP3 and NIX are ubiquitinated. So this is a different flavor of a tag that goes on to these receptors, and it helps to turn the proteins over. So mitophagy is turning the whole mitochondria over, but the proteins themselves can be turned over, and that's like a switch to turn these things on and off. So specifically, if you put a ubiquitin on BNIP3 and NIX, it's going to turn it off because it's going to promote its turnover. So it's been known that BNIP3 and NIX are tagged for turnover for a long time, but we didn't know the regulators of it. And frankly, we didn't know how important it was until a really important set of studies over the last 10 years came out, first linking FBXL4, which is a ubiquitin ligase, to mitochondrial disease, and then linking FBXL4 to actually putting the ubiquitin, or this tag for turnover, onto these molecules. So probably 3 or 4 years ago now, multiple groups identified and linked FBXL4 as the thing that tags BNIP3 and NIX to be able to turn these things over. But it turns out that the model is a little more complicated than that. FBXL4 alone can't tag BNIP3 and NIX effectively. It needs PPTC7 as an adapter molecule to essentially bridge BNIP3 and pull BNIP3 and NIX toward FBXL4 to be able to facilitate that ubiquitination, that tagging. So if you lose either FBXL4 or PPTC7, you essentially lose the ability to tag these proteins for turnover. And that leads to their stability, and that leads to too much mitophagy happening.
These findings that Dr. Nini shares were published in 2024 as a back-to-back publication with Dr. Pagan's group. Another group in China, led by Hui Zhang, also confirmed these findings. This is yet another example of groups around the world coming to the same findings, which is a very valuable way of confirming what we see in our models is real. Around the same time, families like Courtney and Kevin Klein's were stepping forward to share their children's stories.
Courtney and Kevin's story represents just one of the hundreds of parents who have received the stark diagnosis in the last 12 years. Their daughter, Emery, is one of the angels we fight for. Her story is heartbreaking, but also filled with love and determination to keep pushing for a cure. Courtney and Kevin shared their journey with us.
About a year and a half ago, we were pregnant with a little baby girl we knew was on the way. It was, for all intents and purposes, like perfectly normal pregnancy. We had a couple of little scares in some of the ultrasounds that were ultimately investigated and written off. We didn't see any reason to be concerned. And so on the day Emery was born, we went to the hospital totally expecting to welcome a perfectly healthy baby girl into this world. And so she made it through delivery and up to recovery. About 12 hours after recovery, she started making some really weird grunting sounds. And so we had asked one of the nurses to check her out. She took her to the NICU for the evening to monitor. And then that quickly progressed into a very intense next few days of being transferred to a specialty hospital. Nobody really knew what was going on. There was a bunch of doctors in and out trying to figure out what's going on. That turned into a 2.5 month stay at the NICU. And eventually we did find out that she was diagnosed with FBXL4, a mitochondrial disease. At the time, we really didn't know much about it, and we've since learned quite a bit. But we brought her home after the 2.5 months. She's still very sick little baby. But at that point, we had started to make peace with, we didn't really know how long she would be with us. It's just likely only a few months is what we were being told. And so we were intent on giving her the best life we could with what we could. Now, fast forward a year and a half from then, and it's been a very long year and pretty hard. I think Courtney and I have been working harder than we ever thought was possible, and certainly longer than we ever thought was sustainable. And we've really just been focusing to do everything we can to give baby Emery the best life that she can, while also giving her big brother, and she's just got a 3-year-old brother, Weston, the childhood he deserves. And he's awesome. He doesn't, to him, baby Emery is just baby Emery. He constantly makes comments about when he was a baby and he had a feeding tube, or when he was a baby, the nurses would help him do this. And it's totally reasonable for Emery to just be at the doctor's for a couple days at a time without any question. That's just how babies are to him. And so it's really cool seeing her life through his eyes. He's just that pure child innocence and a really good big brother to her, brings her blankets and toys all the time. And so on top of all that, Courtney's been making pretty major strides into directly advancing mitochondrial research. And she's spent a ton of time raising funds for labs and connecting research groups from around the world to leverage their specific expertise. It's kind of crazy she does that, in my opinion, because she's also the one carrying the majority of the mental load of spending hours on the phone in circles with insurance and coordinating all of Emery's care day to day, and making sure we have nurses lined up for the next week. And so it's pretty incredible what she's done. It's beyond what I can do in the medical side. Somehow in all of that, she still finds time to read every research article that gets posted that might be even partially related to FBXL4, and tries to understand what's going on with Emery's condition.
You know, one of the sweetest things about Emery's story is the innocence and pureness that her brother Weston has, and how he sees her and how he relates to her. Kevin and Courtney and Weston share such a special moment. Courtney and Kevin share their favorite memories with Emery.
Yeah, I mean, my personal favorite memory, she doesn't smile very often, so it's always pretty magical when she does. And there was a week where she was in the hospital recovering from pneumonia, and Courtney and I had just traded off. And so it's near the end of her stay, but I was sitting in the room with her, and all of a sudden I just started hearing these really happy baby squeals. And so I walk over to the bed to see what's going on. And she had found a little saline bottle. It looks like an eye drop bottle. And she was holding it in her hand, and she was just squealing with joy. And it was the first time I saw her like truly happy for no reason. And she's not very coordinated, so the fact that she even found that bottle and was able to grasp it and hold it was pretty funny. And so she held that thing for a long time.
It was a baby hand, so like the whole saline bottle was
She's like the perfect size for her hands. And so, yeah, for weeks after that, she was just hold that. She's also got a little toy with a big red button that she can push, and it will play songs and lights. And Weston always brings it over to her. But the first week she got that thing, she was obsessed with it. Like, she would sit there and listen to the music, and then she'd actually intentionally hit the button, which is pretty impressive, given that she's not very coordinated. She can't see very well. And so, yeah, Weston always makes sure to bring her the toys and put them within arm's reach for her. And I think for him, she's kind of the perfect baby sister, because she doesn't take any of his toys, so he can selectively bring her toys and she plays with them.
Yeah, he's right. It's so magical when she smiles, and it's so like you can never force it. Like, there is no smiling. You pull out the camera because she was being so cute 5 seconds ago, and like she won't do it again. She is still, I mean, she's almost 15 months old, so she's still a pretty young baby. I feel like we haven't truly seen, like, Emery just has like a lot more to show us, I think. Like, she's still like a little newborn that we're kind of waiting to see her personality come out. But she does pretty much the two things she loves are, if I kiss her giant cheeks, those are like a pretty high likelihood of smile, or if Kevin makes like popping sounds with his mouth, I think those are like her two things. She's like, that's like how she associates mom and dad. So when she gets her cheeks, and probably she can smell me or something, she'll be like, because she's basically blind. She can't really actually see us, but I think she can tell by my kisses that it's me, and she'll like light you up with just like this warmth. And as soon as she hears him walk in the room and make like a popping sound, she'll laugh.
Hearing Emery's story brings us back to why we fight. Every child's memory, for every family still searching for hope, for the beautiful Emerys and the brave Benjis that are out there who are still living with the complexities of this disease. As a parent, I feel incredibly fortunate that we have so many more resources and the support and understanding of the disease now that we didn't have when I first received Lorelei's diagnosis. And I had a very confusing moment with this brand new baby in my arms, not knowing what to do. Courtney told us what it was like learning about her daughter's disease.
I think the information's out there, and hopefully, like, I mean, this podcast is a perfect example of, I hope we have more and more things that are readily easy to understand, because I think knowledge is power, especially when things feel so out of control for your kid. Like, if you can at least understand what's going on, you can, like, at least for me, I could find, like, I can actually come to peace with her disease a lot better when I can explain to myself what's going on. And I think when she got her diagnosis, I questioned if I would ever understand what's going on. And I think we know a lot more about the disease than I realized at that time. And so I think if you're a new parent and you want to know, like, use all of your resources. Reach out to the FBXL4 parent community. There's a lot of us who have thrown ourselves in. Michael's another good example, who are willing to catch people up. And I've had a lot of other parents reach out to me and want sort of the intro, because they also want to start reading articles. I've had a couple parents reach out and say, like, what's the best first article to read? And I'm hoping now that this podcast comes out, I'll say, start with this podcast and then go read.
I think part of
those other two.
Part of that's pretty important too, of like, it's such a rare disease that the doctors most of the time have no idea. Like, if we go to a different hospital that we need to go into the ER or something, they have absolutely no idea. And they see Emery and her labs are all off the charts and they want to immediately start intervening. And we're like, this is her baseline. And so I think having that community is really important to not only help gain knowledge for yourself, but also your local doctors, and being able to feed that information back, because there is a lot of different work around the world that's going on. A lot of different doctors are trying different things to help. And so knowing what's going on is also really important, because we've had those conversations with Emory's doctors of like, hey, this hospital's doing this. Have you talked to them? And they'll go and consult, or this research group is getting involved in this. Is it something we should also pursue?
As a parent, I totally agree with Kevin. Because this is such a rare disease, oftentimes, especially when you're walking into the ER, you have to be prepared and talk to the doctors about what's going on. As scary as Google is, and I don't recommend Googling things, but we all have to do it. Parents are doing our research, and we're talking to our team to help care for our child in the best way possible. As we said before, each child is so unique, and this disease presents differently. And connecting with other families is one way that we can continue to support our kids.
And for us in the science world, this is a reminder that every experiment we do is ultimately about these kids that we get to help.
We say all the time, like, we're like, Emery, you just want to be a baby, don't you? Like, all you want is to be a baby. And we'll see her hooked up to all these machines and doing all these therapies and nebulizers or whatever, but all she wants is to hit her big red button and grab some crinkle paper, and that's all she wants to do, and be snuggled.
I put her on your chest, and that's her happiest place, to just lay there totally content for hours.
So listeners, I thought I'd quickly summarize where we are and what we know about FBXL4-Associated Mito as of the fall of 2025. FBXL4-Associated Mito presents with a range of severe symptoms due to an uncontrollable removal of mitochondria. Basically, cells have evolved a way to target damaged mitochondria for removal. This process is known as mitophagy or mitophagy. Mitochondria have receptors, BNP3 and NIX, which signal for the mitochondrion to be removed. To protect healthy mitochondria from removal, our key proteins, FBXL4 and PPTC7, bind to BNP3 and NIX to degrade them. This preserves healthy mitochondria to produce the energy we need. However, in disease, healthy mitochondria are removed. This is because when FBXL4 and PPTC7 are mutated, BNP3 and NIX accumulate and signal to remove healthy mitochondria, meaning that there are less mitochondria to produce energy, which subsequently leads to FBXL4-associated mitochondrial disease. So where does this leave us? FBXL4-associated mitochondrial disease is rare, but if you've stumbled upon this podcast from searching up that crazy word you've never heard of before, we hope you realize that you are not alone in this. Families are fighting, scientists are fighting, and we're starting to fight together.
All too often, these battles are happening in isolation. Families don't always know that there are labs full of scientists working for them, and scientists don't always see the children that their research serves.
But when we connect, when families and researchers look each other in the eye, when we do podcasts like this, we're creating something stronger than just data and grief. We're creating hope together. For families like mine, it brings comfort to me knowing that it's not just me and my husband who think about FBXL4 on a daily basis. There's whole teams of scientists doing the exact same thing, all towards finding a cure for kids like mine.
And that brings us to the heart of this very initiative, bridging the gap. And so we asked,
what do you wish scientists or families knew about your FBXL4 journey?
I think it's great when researchers can connect with the families. I think it's important to have that why and to see the effect that you're having. You know, if when Lorelei was born, I remember like asking about FBXL4 and what it does, and I didn't really get a satisfactory answer. And it turns out like it really wasn't known, right? It was involved in mitochondrial DNA maintenance, but what exactly does it do? And then I think it was in 2023, I think is the first paper that I saw where they talked about the BNIP3 and the NICs and how it degrades those receptors. And so starting to get a better idea. So when a researcher, figures that out, instead of say, hey, I figured out what a molecule does, versus, we're one step closer to helping, find a treatment for this family. So I think it's great to be able to, for researchers and families to be able to connect. And I think I like how UMDF does their conferences too, where they have the researchers and the families there at the same time. That was, I don't remember if it was 2016 or 2017, but right after
I think you went to 16,
Seattle,
17, and 18.
Yeah, I went to, trying to research mitochondrial disease and flew to Seattle, went to the first Mito conference and learned a ton from somebody who doesn't, I didn't even know, the disease exists. And so, yeah, that is not really what I wish the researchers knew, but I'm just glad we have that connection and that ability to do that.
I wish that they knew, first of all, that we're grateful for everything. It's really comforting to know that there's somebody in this world who thinks about FBXL4 more than we do. And like we do this every day because of our kids, but we think about it differently. And it brings comfort to know that there's somebody thinking about this tiny little disease that, there were 3 case studies for 10 years ago, and that there are people who like think about this every day. But also I would love for researchers and scientists and even doctors to know that our kids are our kids. We know them best when it comes to like them specifically, but also that they're kids, that they aren't science projects, that they may act different and they may not be able to sit up, but they are still kids and they still bring joy.
So what I would like patients to know, we can say that now we know the pathophysiology of this disease. We slowly know more and more about this protein. And by knowing more and understanding in which pathways or where this protein is working, I guess in the future, potential therapies could be developed to counteract what happens in this disease. So that would be what I would say to the patients. It's a process that might take, of course, a lot of time to develop a therapy, but by knowing more and more about the pathophysiology, this gives hope to these patients, I guess.
You guys offer families hope that we don't otherwise have.
Yeah.
Before Emery being diagnosed with FBXL4, we didn't know about it. Nobody in our family knew about it or friends knew about it. And then even after that, like Keri said, there were very few, like the Google searches didn't turn back very much. And so we were leaving the hospital with a very sick baby with very little information and really not much coming out of the doctors other than bring her home, give her a happy life. But then the more we've gotten involved with the research community, the more we've realized there's a ton of people who are dedicated to this cause and potentially have a path forward to, maybe they don't help Emery's specific case, but maybe the next kid doesn't have to go through the same struggles that she's had. And so that's a level of hope that the research community offers to families that I think is really important to focus on.
I can't tell you the number of times you could quote me from saying like, it's an entire PhD and like her whole job is my kid's disease, but like, it's not just one person, it's like 10 people. And it's like, they're literally PhDs who their whole 40-hour work week is my kid's disease. And when I tell people that, they're like jaw drop. And that's when I think light bulbs start going off people's heads of like, they finally believe me when I say things are happening. Because I think a lot of rare disease, every rare disease parent wants to believe things are happening. But when I explain what's actually going on in this specific disease, I think people are like, oh my gosh, you're right. Like things are happening. And we're so glad for it.
So if I could say one thing to patients about our work, it would be to explain that we deeply feel that research is very slow and very frustrating, and especially for people living with conditions and desperate for treatments and answers. We know that there's an urgent need for us to do the research, but the other side of that is that we have to make sure it's safe and solid so that we don't give false hope to people that are already bearing the burden of mitochondrial disease. Patient stories matter deeply to us. It really motivates my team and I. Without these stories, we feel we're just doing experiments in the lab. So we really think that hearing from patients helps us see the bigger picture.
Yeah, so this is a great question. And one reason why I really appreciate the UMDF and the mission is because as a researcher, it's very impactful for me to be able to connect with and see the stories of the patients who have mitochondrial disease who might ultimately benefit from our research. So I think that the thing that really I hope patients know about our research is that we see our work as part of a team effort. So we're performing research at a very basic level to try to make discoveries that ultimately could help us to understand mitochondrial disease and help clinicians and doctors diagnose and treat mitochondrial disease more effectively. And this, together with researchers and clinicians, will ultimately allow patients to live healthier lives. So I think it's a team effort. Researchers, clinicians, patients, families, all of us working together to try to benefit and push forward the knowledge about mitochondrial disease.
The diagnosis and the progression of mitochondrial disease is truly devastating and exhausting for both the child and the parents. And knowing that there are really superheroes in lab coats trying to find a treatment, cure, even the mechanisms of how these genes work and how to better allow them to function. I'm just so grateful because I do think that within my lifetime, we will see at least therapies that will help the mitochondrial disease community. And that is not possible without these brilliant scientists. So for me, I am so grateful. I'm so grateful.
These voices remind us that none of us are in this fight alone.
When families and scientists connect, we create something stronger than data or grief. We create hope.
And this is just the beginning. The story of FBXL4-Associated Mitochondrial Disease is still being written. One discovery, one child, one connection at a time. A special thanks to all our guests and families who have contributed towards this podcast. Specifically, I'd like to thank Susan and Mike Aiken, Courtney and Kevin Klein, and Janice Belcher for sharing your kids and your favorite memories and all the lovely stories with us about your journey with FBXL4. I'd also like to take this opportunity to thank all the families that send through photos of their kids. We really appreciate just being able to be a part of your journey and your story too. I'd also like to thank Dr. David Alsina, Dr. Julia Pagan, and Dr. Natalie Nemi for sharing scientific insight with us and paving the way towards a cure for this very rare form of mitochondria.